Continuing children with our last topic of polyembryony. Let us see by definition what it says. Development of more than one embryo in a seed, and the process is called polyembryony. Let us first go back to our previous diagram here where we discussed about double fertilization and the triple fusion. How is the embryo formed, children? This egg cell in the egg apparatus, when it fuses with the male gamete, the process was called syngamy, and this diploid zygote that is formed as a fusion product, it further grows into embryo or the baby plant that is made up of plumule and radicle. So I hope I have reminded you that how is the embryo formed in an embryo sac, right? So coming to our current topic, we are talking of polyembryony. So when we say poly, we mean to say many, and we are talking of a situation in which many embryos are formed inside an embryo sac. So who discovered this first? Leeuwenhoek in the year 1719 in oranges. There are two types of polyembryony, true and false. In the true polyembryony, what happens is that extra embryos arise in the same embryo sac in which zygotic embryo is developed. That is, this egg cell by the fusion of male cell of gamete forms the zygote, and extra embryos will form inside the same embryo sac, not one but more than one, but in the same oval embryo sac. Then you'll call it as true polyembryony. But here, if you talk about the false polyembryony, in this case, more than one embryo develop in different embryo sac but inside the same ovule. So look at this comparative diagram, children. Here, extra embryos are formed in both. That's the similarity. That's why polyembryony. But the difference is extra embryo will be formed inside the same embryo sac in the ovule. But here, ovule will be the same, but embryo sacs would be different. And that's the meaning when we say false polyembryony, that more than one embryos are formed or developed in different embryo sacs inside the same ovule, right? Now, if we talk of the polyembryony, how it develops in seeds by different methods, then we have 6 methods as the answer. The first, it says cleavage polyembryony. Second, embryo when it is formed from synergids. Third, from the antipodal cells. Fourth, from the endosperm. Fifth, what we call adventive embryony. And sixth and the last is from other embryo sacs in the ovule. So let us see the first one, cleavage polyembryony. The zygote, which is usually formed as a product of fusion of egg cell with the male gamete, that undergoes cleavage or division, and then more than one embryo is formed. Example is seen in both angiosperms and gymnosperms. It's very commonly found in gymnosperms and in angiosperms in plants like Nicotiana rustica, Nymphea advena, etc. Coming to embryo from synergids. Now, what are synergids? These side 2 cells next adjacent to the egg cell. So, these synergids may be fertilized by male gametes and they develop into an additional embryo. Example, Sagittaria. Third, embryo from antipodal cell. What was an antipodal cell? Those top ones. So, those antipodal cells may develop into extra embryos. For example, Ulmus americana. Then embryo from the endosperm or the food part that is stored therein. Then any one cell of endosperm may grow into embryo. Example, Belanophora. The fifth is adventive embryony. This is seen in plants like Eugenia and Mangifera indica, that is scientific name of mango. Embryos develop from diploid sporophytic cell of the nucellus, the nutritive tissue of ovule, other than zygote, which penetrate into embryo sac. And finally, the last one, from other embryo sacs in the ovule. Example, in citrus, that is from any other embryo sac in the from other embryo sacs in the ovule. Example is citrus. I'll show you by the diagram here. This is polyembryony in citrus. You can see this is the integument, the nucellus. Then we have the zygotic embryo, endosperm, then the nucellar embryo, and that's the integumentary embryo. So I hope you've understood, children. With that, we come to an end of polyembryony and obviously the end of this chapter. And before we close this, I'll also show you where we are in the textbook. With this, we complete with lesson number 2. My next video will be on the 3rd chapter that we'll start, children. And please go through the topics very clearly. Here, just to close, I'll show you the true and the false fruit that we did today. This is the fruit of mango and coconut. Please practice the diagrams and do them as a homework. Label all the parts, the epicarp, the edible mesocarp in mango, and look at the mesocarp in coconut, which is fibrous, which I explained to you. Both the fruits we call as drupe. Yes. And then here we have the false fruit. This is the apple. This is the thalamus part. As I told you, we have the thalamus as the edible part. And then this is the strawberry, where this is the thalamus. Then this is the mesocarp and the endocarp. Please practice a few diagrams, especially apple you can draw and practice. And when we have the true fruits, practice mango and coconut. With that, children, we come to an end of lesson number 2. Go through and ask me any doubts if you have, and keep learning. Thank you.